Yeah, there does seem to be quite a few tragedies going on around the world at the moment. Australia was hit very hard with floods, actually, our area, as I was just saying to Denise before we started. And I think you are right, Denise, in talking about, how our environment is stretched, we're stretched, the way we're living our lives, the way we're treating the world around us. And when we talk about this, this topic tonight, obviously we're talking about health span and vitality. But, Denise, you use the word resilience, and I think that is a really important word, particularly with what's going on with all of you right now. And even for those that aren't experiencing floods or these kind of things, being resilient is such an important thing. And resilience comes from being mentally strong, physically strong, having a good immune system, being able to adapt, going through something that is challenging or even traumatic, but being able to adapt to that situation, move on, possibly even grow and learn and become a better person in that kind of situation. Whereas some people do not have that ability to be resilient, are not able to adapt, are not able to grow and deal with whatever's going on. And we're going to talk about epigenetics tonight. When someone does experience stress and trauma, there are epigenetic changes. These methyl groups sit on the DNA, literally change. And we need to be able to undo that so that these traumatic experiences don't literally stay on our DNA, turning on certain genes that are going to increase our risk for mental health disorders and chronic disease, because we do know there are a lot of studies, particularly around chronic stress and trauma, increasing, mental health and chronic diseases. And that is through the pathway, that mechanism of epigenetics. So the topic for tonight is health span, as Denise said, a relatively new term for some. So health span is really talking about our time on this planet where we are healthy. So it's not so much about living longer, but it's about increasing our health span. So I'm not sure what the health span is in the Philippines or Hong Kong. If anyone knows, feel free to put it in the chat or share it with me at the end of the talk. I do know in Australia, the health span, based on data, this was published in 2020. We don't have anything newer than that at the moment that I'm aware of. It was published in the Lancet. And actually, it's probably got some data from Asian countries in there as well. I think it was 19 countries they published on. But in Australia, our health span is 70 years, but our lifespan is 82 years on average. So on average, we have 12 years in this country where people are living with a chronic disease, some kind of disability, are likely in pain, possibly having issues functioning, things like that. In New Zealand, it's pretty much similar. I think the health span's about 69 years, living to about 81. And I do know globally, so the United Nations very recently published that globally, I think the health span, lifespan is about 73, so a little bit lower than us in Australia at 80. But the United Nations has said globally, lifespan for all countries together, on average, people live to the age of 73. But most people, according to United Nations, have at least 9 years of a gap, a health span gap, where they are struggling with disease and health conditions. And I'm pretty sure that most of you listening don't really want to have 10 years of struggling. So I've seen my parents go through their health issues, and that really does stimulate a lot of the work that I do. So I'm an advisor for Pure Encapsulations, and I've got no other conflicts of interest to disclose. And tonight we're going to talk about methylation, epigenetics, and aging. We're going to talk about the effects and influences of certain things on epigenetics and aging, or factors, things that we can influence as practitioners to help people improve their methylation status, reduce their biological age so they have an increased health span, not just lifespan. And we're going to chat about a current biological aging study that I'm doing that Denise is also part of. So before I launch into the epigenetics, I just want to chat a little bit about genetics, or your DNA, your genome, and what makes you uniquely you. So every cell in your body, every cell with a nucleus, contains the recipe to make you, you. So we obviously inherit our DNA from our parents, and this influences our phenotype, the way we look, our height, our eye color, where we distribute our weight, if we're able to produce lactase and tolerate lactose and dairy, if we're likely to be more sensitive to gluten due to HLA genes. Our genes really do influence not only the way we look, but also our susceptibility to health and disease, our dietary preferences, sensitivities, etc. But it's not just our DNA and our genome that is important. We also want to consider our epigenome. So when we talk about our epigenome, we are talking about literally things that sit on top of that DNA. So that instruction manual to make you, your genome, that is made up of 4 bases, A, T, C, and G. And basically, your genes need to be turned on to make proteins, whether they're structural proteins, whether they're going to be hormones, neurotransmitters, transporters, enzymes, etc. But what regulates turning those genes off and on is the epigenetic component. So epi literally is the Greek prefix for meaning on top of or above. And epigenetics, as I said, it's this additional layer of instructions that controls what genes are turned off and on. Epigenetic marks are robust, but they are not fixed. Now, 20 years ago, when I first started learning about epigenetics, I actually did think they were fixed. I thought, we inherit actually epigenetic patterns sitting on the sperm and on the egg from our mother and our father. So not only do we inherit our DNA, we also inherit some of these epigenetic patterns from our parents as well. So if your parents are overweight or smoking or older or exposed to chemicals and toxins, actually even stress and trauma can be passed on epigenetically from generation to generation. But when I first learned about it, as I said, which would have been more than 20 years ago now, I did think it was something that was sort of inherited and basically passed on. So every time a cell replicates, not only do you get new DNA replicating, that epigenetic pattern is replicated. But we now know that this is not necessarily the case, that our environment, our diet, meditation actually has quite a powerful effect on epigenetics, can change this. So it's something that we can change. So unlike our DNA and our genome, which we inherit, we can't change, we can change our epigenome. So tonight I'm really going to be focusing on methylation. And the reason I'm going to focus on methylation is because, one, it is the methylation mark or modification that we know the most about. And it's actually something that I studied back when I was doing my PhD and postdoctoral fellowship. Denise said at the start, and I think many of you know me, I've done a few talks now, but my area of interest is sort of fertility, autoimmune and thyroid and healthy aging. So the first 10 years as an academic, I worked in the fertility space, reproduction and pregnancy health, looking at DNA methylation and nutrigenomics and genetics. But I've had actually an autoimmune disease myself, thyroid autoimmune disease that I've recovered from. And my mum actually developed early onset dementia. And I do have the ApoE for allele, the risk factor for that. And my mum actually started getting some symptoms, in her late 40s, and I'm going to be 45. She was diagnosed and very much the onset of Alzheimer's had kicked in by her early 50s. So that is the trigger for me. And then obviously taking on care of my father, the trigger for really wanting to understand this and what I can do to help take control of my health and try to reduce the risk of me having a shorter health span, living a long time, because my mum actually lived for a long time with Alzheimer's, in that state, because, I'm an older mum. I want to see my grandkids, all the things that I'm sure many of you listening want to do as well. So aside from DNA methylation, which I know quite a lot about, I've worked in this space over 20 years, And as the medical literature, the scientific body as a whole, we know a lot more about this than we do histones, and particularly our microRNAs or non-coding RNAs. But just for completeness, when we are talking about epigenetics, I do want to let you know that methylation is not the only epigenetic or DNA modification that affects gene expression, but methylation is the major one, as you'll see as I go through the slides. So I mentioned at the start, we've got our DNA that has our genes. These code for all the proteins that make you, making up your cells, tissues, organs, obviously influencing your health, your energy, your day-to-day functions. But it's not just the genetics, it's also the epigenetic factors that are important. And we know that altered protein function, whether that is, as I said, an enzyme or a transporter, a structural protein, whatever it may be, that can happen if we have genetic variations or mutations, but also if there are epigenetic changes. And often they can occur from environmental, insults. Denise said at the beginning, toxins and things like that, we know they have big impacts on epigenetic patterns and gene expression. So when we talk about DNA methylation, as I said, these methyl groups sit on the DNA. If you can see this image here, this is meant to be a DNA strand, and instead of it being a DNA double helix, it's stretched out like a ladder. We can see our DNA code, A, T's and C's and G's. Methyl groups attach to a cytosine, a C. We have these, what we call CPG islands. A CPG island is literally, it's just called a cytosine, and then we have a phosphate bond and a guanine. But we have these CPG islands, lots of C's at certain regions of the genome, and the methyl groups will attach here if a gene is meant to be switched off, or if for some reason there's an environmental trigger or a stress or something that changes gene expression, and then that gene will become methylated. So when it comes to methylation, it's S-adenosyl methionine. Some of you may give SAMe as a supplement. That's a methyl donor. So SAM or SAMe, if you're giving the supplement, provides these methyl groups. And a methyl group is simply a carbon and 3 hydrogens. And when we say the word methylation, literally, technically, methylation is just adding a methyl group either onto DNA or protein or a lipid, whatever it may be, and then there's sort of a downstream effect. But from an epigenetic perspective, we're obviously focusing on DNA methylation, not so much the methylation that can be occurring if we're talking about breaking down, neurotransmitters. So, for example, for those of you who are aware of, COMT, catechol O-methyltransferase, that's an enzyme that picks up a methyl group and adds it onto something like adrenaline. So if we're stressed, that can help us, de-stress, break down our catecholamines. So methylation is just the process of adding these methyl groups. And when we talk about epigenetics, we are specifically talking about methyl groups being added to our DNA. So just to reiterate what I was saying about methylation suppressing or turning genes off, this is to help you understand that. This black line here is sort of representing that DNA strand, and then the blue box is representing a gene. So just in front of the gene, we have this promoter region, and what would happen is we have certain factors known as transcription factors that bind, and that starts the gene expression. We get, certain things happen, and then the gene is read, and then we make a protein. In the case of methylation, though, it actually blocks the ability for these transcription factors to bind, so the gene is not turned on. So a bit of a fun fact, there are approximately 29 million locations in the genome that can be methylated, and it's different in every single cell. So pretty complex and pretty amazing when you think about it. And it has to be, this complex and I guess this abundant, because when you think that every single cell in your body contains your entire genome, we need so many of those genes to be turned off. Because, differentiation is really a prime example here of methylation and epigenetics. So the reason methylation is so important, and we do have so many sites on the DNA that are methylated and it's different every cell, is because cells have to behave differently. So looking at this diagram, if we have a fat cell, we know that all of the genes that are not linked with the function of a fat cell are turned off. They're all methylated, whereas the genes that are essential for the function of the fat cell are not methylated, so they're turned on. Here, looking at epithelial cells, obviously all the genes are turned on are in relation to the function of epithelial cells. So you're not going to get, for example, insulin being expressed. That would only happen in a pancreatic cell. So methylation is really important for suppressing all the genes that should not be turned on, and then only the genes that should be turned on. And by the way, this is assuming that the body's functioning properly. Sometimes there is imbalances and changes in methylation. That's when we start to develop chronic conditions, autoimmune disease, etc. But methylation is really important for ensuring that the right genes are turned on at the right time. And just to bring all this together, sort of thinking about epigenetics and methylation before we move on to the next section. So, here we can see we've got a chromosome. Chromosome is basically made up of tightly packed DNA. The DNA is wound around what we call histones, and together that DNA and that histone is called chromatin. So when the DNA is tightly packed, we obviously can't get those genes. Those genes can't be turned on. So what happens if we need to turn on a gene? We actually have acetylation. Now, I haven't talked about that yet. It's not the focus of tonight's talk, but just know that the other factor, one of the epigenetic marks, acetylation, is what's involved in turning genes on. So acetylation kind of unwinds that DNA so that it's open, transcription factors combined, and then the right genes are turned on. Whereas with methylation, as I said, not only does it block genes being turned on, it also promotes that DNA being tightly packed and closed so that transcription factors can't even access those genes as well. So we have these factors, acetylation, methylation, and even microRNAs that directly influence what genes are turned off and on. So we've talked a bit about methylation and epigenetics, and now I want to focus on the relationship with aging. So aging is basically a time-dependent functional decline at the cellular level. I think we all know what aging is. We start to get wrinkles, we go grey, we've got, degeneration, inside the body as well. And we know that aging is the primary risk factor for chronic conditions such as cancer, type 2 diabetes, cardiovascular disorders, as well as neurodegenerative diseases, Alzheimer's and dementia. So when it comes to aging, there are a number of factors that have been identified that basically change, that alter, that trigger these, this cellular dysfunction, and that's really what happens with aging. So we have genomic instability, and this is an area that I've worked in as well. So when I was looking at methylation, I was also looking at DNA damage as well as genetics, telomeres. So in my lab, I actually didn't test telomeres. I'm doing a bit of telomere testing now through this commercial test that I'll tell you about. But when I was doing research and had a lab, even though we had the ability to test telomeres, that wasn't part of my work. Epigenetics, which is obviously a big factor, as well as, mitochondrial dysfunction, stem cell exhaustion, etc. So when it comes to epigenetics and really predicting age, or more so biological age rather than chronological age, because there is a difference. So chronological age is, how old you are, thinking about when you were born, how many birthdays. So for example, I am about to be next week 45, but my biological age, maybe I'm 40, maybe I'm 50. I will disclose I'm actually older, but I did think when I started this research study that I was going to be older. And actually, as I said to you all, what sort of driven me into this healthy aging space is because of watching my parents suffer, but also knowing, like Denise said, that I have, I guess, even though I have all the knowledge, like many of you, on diet and exercise and sleep and meditation and reducing toxins, there are some things that I'm very good at. I'm very good with my diet. But, stress is an issue. I've had Graves' disease, a bit of stress last year with my dad passing. There's been multiple things. We all experience it. Many of you are experiencing stress now because of, what's going on around you. We can't avoid stress, but we want to be more resilient and take care of ourselves so that our biological age is reduced and our health span is increased. So we're not living in a state where we are unwell. But this diagram here is actually something I've been using this slide I've used for a long, long time. It's from a paper published back in 2013, so 10 years ago, before we were really even before I knew about predicting age or sort of these epigenetic aging clocks. But we've known about this epigenetic drift. So what we can see here, this middle line, this is representing normal aging. So down the bottom, we've got our chronological age, and then we've got our biological age. So think of biological age as more like your cellular age. You could be older or younger, depending on your exposures, how well you've taken care of yourself. So some people can experience premature aging. This is the red line here, if you can see my mouse. And this is actually a term that I would say in 2023, we don't tend to say premature aging, we tend to say accelerated aging. So accelerated aging occurs due to, exposures, smoking, obesity, stress. There are a number of factors. Alcohol is one of those things as well. High blood sugar. There are a number of factors that can increase accelerate our aging. And then we can also have what we call healthy aging, where we have a slower pace of aging. And that slower pace of aging is really what we're going to sort of talk about tonight and what we're aiming for in our biological aging study. So epigenetic biological age is an age estimate in years based on methylation status, based on the methylation or the methyl groups attached to the CpGs, those cytosines in your DNA. And we can test that now to get an assessment of your biological age. It relates to your cellular health, and it's a better predictor of your real age. So even though you have an age, as I said, you might be younger or older. Epigenetic aging can be reversed. Yay, thank goodness. That's what I'm aiming to do. And DNA methylation is a promising marker for studying human development, aging, health, and also for evaluating rejuvenation therapies. And that's really what this study is about. We are evaluating certain supplements and protocols to see if it can actually reverse our biological, or at least slow our biological age. So with our epigenetic biological clocks, DNA methylation has been strongly correlated to age and age-related or health outcomes, age-related disease. It's been shown to be highly accurate, and we know that it changes with lifestyle and time. So 60% of our methylation patterns are non-heritable. So there is a heritable component. And not that I'm making excuses for my older biological age, but my parents were older. Mind you, I was older when I had my kids too, but my dad was 40, he smoked, my mum didn't. At this stage, when we look at the research, smoking in pregnancy for women actually does have epigenetic changes that are linked with accelerated aging, even in the infant and the child. So accelerated aging or slow aging can happen at any time point. It's dynamic. It can go up and down. My mum didn't smoke, but she was exposed to, a heavy load of secondhand smoke. And back in the 70s, that was normal. People smoked inside, in the car with the window up. Obviously, things have changed a lot since then, though. So over here on the right, this is an example of, a part of the testing that we're doing actually gives you lots of different age estimation clocks. But this one, just looking here, this intrinsic age, what we can see is this person, and this is actually the oldest person in our study. So when she did the test, she's now 82, but when she did the test, she was 81, almost 82. But her biological age actually came in 9 years younger. Now, I know this lady. I have been working with her daughter, and she's very healthy, by the way, in her 80s, but she's cooking, she's independent, she still goes for jogs. She does blood testing every year. She also does some functional tests. But as I was saying, I've been working with her daughter now for over 10 years. And when her daughter sees me for healthy aging, basically she always says, I'll also get this test for my mum, and I'll get these bloods for my mum. And she actually enrolled her mum in the study as well. So it's no surprising that she is significantly younger, because their diet is excellent. As I said, low exposure to chemicals and toxins. She does work on her sleep. She's active, all of these things. So just a bit about the history and sort of the background, the research behind these epigenetic clocks. So the first age prediction linking with DNA methylation was published back in 2011 from data from the UCLA. However, we have known about the association, so not so much predicting age, but the association of methylation and age for decades prior to this. But this was the first publication trying to predict age with methylation testing. Then Stephen Horvath came in. So he's actually has one of the most predictive aging clocks. And he found that different tissues actually age at different rates. So for example, breast tissue ages at a faster rate, our brain tissue ages at a faster rate, but the immune cells seem to, in most people, age at a slower rate. Cancerous tissue, on average, has been found to be 36 years older than other tissues. So there is definitely accelerated aging in cancerous tissues. So there's been a number of different age estimators over the years. I will sort of walk you through some of those to help you understand where we're at in 2023. And we know that these epigenetic clocks can predict biological age more accurately compared to chronological age. And as I've mentioned, it can be reversed. And in addition, it's not just about trying to predict an age. We know that DNA methylation is also strongly associated with these age-related conditions and can predict lifespan and health span. So the testing that we are doing is not predicting lifespan and health span. I don't know if any of you listening know some of these aging clocks. There is one called GrimAge. GrimAge actually predicts how long it expects that you will live. As functional medicine practitioners, we don't really want to be telling people when they're going to die, but also the people that produce that have painted anyway. It can't actually be used, but it has been shown to be quite predictive. But in this case, we're more interested in, function and not to do with lifespan. So just to walk you through a bit of the history and where we're at now with looking at methylation and aging, there were these first generation clocks. So this is the first one that I mentioned. This was published back in 2011. But there were a number of publications, epigenetic aging clocks that were produced by different groups over the years. We then had these second generation clocks that were called, they were trained using aging phenotype. So they weren't just necessarily trying to predict age, they were also then looking at blood markers. So bringing in biomarkers, and these were a bit more predictive of age, but also of age-related conditions and health as well. And what we have now in 2023, and no doubt over the years, if we keep talking about this, there'll be fourth generation, fifth generation, but what we have now is these third generation clocks. And these are highly predictive at telling us about our pace of aging. So what's different about this this what we call the Dunedin pace, this pace of aging, is this is not predicting a number. This was not trained, because there's a lot of bioinformatics and algorithms and things like that. This was not trained trying to predict a number. This was actually trained looking at functional markers, things like grip strength, which is known to be associated with aging, the one-legged balance test, MRIs, cognitive decline, facial aging, blood biomarkers, as well as outcomes like cardiovascular disease. Also, it's a longitudinal study. It's still going, time of death as well. So this is basically probably one of the most accurate when it comes to function. So where someone's at right now, their pace of aging. Do they have a slower pace of aging or a faster pace of aging? So when we do these tests, and the one that I've shown you, and I'll show you a few more examples, we do get an estimate of age, biological age. We get some estimates around immune health, but we also get this pace of aging. So you may not want to get stuck in the data. I'll skip over this, but if you are really interested in the comparison with Dunedin pace and the other clocks that are out there, if some of you already have some knowledge in this area and you want to get stuck into some of these papers, you might be interested in this. But this is really just showing that they have compared the Dunedin pace, this functional marker, with these other epigenetic aging clocks, and it does seem to be more predictive in relation to mortality, cardiovascular disease, stroke, as well as a number sort of functional or daily living activities, including things, such as pushing and pulling, people being able to dress themselves, etc. So a good idea or a good association with function. So the Dunedin pace, which is this test that's looking at the slower or accelerated pace of aging, has been strongly associated with health span as well as endpoints, like I said, cardiovascular disease, etc. And it adds an incremental prediction above and over all of these clocks. So the testing that we're doing in this research study and what's available is we're actually looking at multiple epigenetic clocks, aging clocks. So it's not just one. I showed you one part with the biological age. We're not just looking at one. It has pooled information, the best epigenetic clocks that we have at the moment, including information on telomeres and other factors, to give people a bit of an overall picture of where their health is at. So factors that influence methylation and aging are obviously diet. The most research we have is around caloric restriction, but what we have to emphasize to our clients is it's not just calorie restriction, it's calorie restriction without malnutrition. So what we need is for people to be eating good quality food and make sure that if they are going to reduce their food intake, they're still getting plenty of nutrients. So when we talk about having a healthy genome, a healthy epigenome, and even for this protein expression, cofactors, we need minerals, we need B vitamins, we need things in our diet. We'll talk about some of the factors, in supplements as well, but our food literally talks to our DNA. Sulforaphane, things in, blueberries, we talk about blueberries being antioxidants. They literally have epigenetic factors. They go in and turn on, detox enzymes and antioxidant enzymes. So basically, this is one of the most important things that we can share with our clients. There is very, very good data. Time-restricted eating is sort of classed as caloric restriction in making sure that people are just having at least a good 12-hour overnight fast. I tend to suggest for women 14 hours, in some cases men even 16 hours if we're trying to get certain endpoints. But in our study, we did 12 hours. We've got 60 people. I didn't want it to be too restrictive. And we've gone 12 hours, and we do give some dietary advice, as I'll share with you. The Dunedin pace that I told you about, they've done a study. This is a very new data, and they've found that with a 12 and 24-month follow-up from calorie restriction, we can see that the pace of aging, so the top blue line is eating what they want, normal diet, and then this sort of orangey red down here, these are those that have a part of that calorie restriction group. And we can see there is a significant decline, so a slowing in that pace of aging. It does seem to plateau out a little bit more, almost go up a tiny bit. I did question this. So TrueAge, the company that offers the test, I spoke to them about this. I said, why isn't it getting better after 2 years? And the explanation that I got is that it's likely that at some point, and this does make sense, there will be benefit when you do things. If you give a certain supplement, you change diet, you reduce chemicals and toxins that can have harmful epigenetic effects. But there is a point where you cannot continue to go backwards. You won't go back to being a child or a stem cell. There will be a limit to these changes. So at this point, it looks like that is the reason why there has been this plateau and not the sort of this continuation, because there's a limit to how far we can slow our aging. So exercise is really important. And if we are talking about aging, health span and vitality, if we want to improve our health span, we need to be strong. We need to be, we don't want to be weak. We don't want to be frail. We don't want to, fall over and break a hip. So strength training really does help slow the aging process, because part of the aging process, when we talk about cellular dysfunction, is sarcopenia. We lose that muscle mass. So strength training at least 3 times a week is best for most people. In addition, if we're just talking about body composition, not just, function and balance and strength, it's going to having more muscle will increase metabolic rate. And as I said here, less risk of injury, better balance, and higher energy levels. So for that vitality side of things. There has been a study, this was published in 2021 in males that were 65 years and older, and it showed that resistance training, so lifting weights for 6 weeks, increased lean tissue and DNA methylation changes that were similar to a younger or youthful male. And I wanted to highlight this as well, because sometimes people think, well, I'm a certain age, I'm 70, we've got a few older people in our study too. I'm not really going to put on muscle, but the reality is you can put on muscle at any age. You will be slower to put on muscle than what you would have been in your 20s and 30s. After our 30s, it starts to slow down. 40s and 50s, it's harder, but you still can improve. So there is no reason not to, make changes. Even if someone's 75, they can start doing these things and get the benefit. And actually, if someone hasn't been training, they tend to get the benefits a lot quicker than, if you're in a space where you're doing a little bit and you want to have a big change, the body doesn't adapt as quickly. But if you've been doing nothing and then you start, as long as you obviously start slow, we don't want people to overtraining is not okay. So when we're talking about longevity, health span, vitality, rest is important, and not pushing ourselves. Overtraining is definitely not the way to go. So what I find with a lot of people, particularly in this aging space or working in functional medicine, a lot of people already have chronic conditions, they've already got pain. There is always something that they can do. And even personally, stretching is something that I have to do a lot. Maybe I don't do it enough. The reason I'm even standing with you tonight is because, the sitting for me is a problem. So I've got some bulging discs, I've got multiple issues. I did overtrain in my younger years. And, a lot of those things affect me now, even though I'm only in my mid-40s. But in saying that, I still play netball, I still train, but I have to take good care of myself. So there's always something that people can do, whether it's a stationary bike, even getting them just to start moving. And in our study, we actually say to people, just move for 30 minutes a day. We have had a lot of education. My co-investigators are an exercise physiologist around strength training. We obviously emphasize that, but the main thing is moving for 30 minutes per day, most days. So when it comes to supplements too, if we're thinking about, improving health span and energy, there have been a number of bioactive compounds or nutrients that have been shown to improve health span or longevity. But mind you, though, most of these studies are in either yeast or, cell culture, worms like nematodes, for example, with sulforaphane. And I think for those of you who've come to my other talks, I've given some examples. We've gone a bit deeper into sulforaphane and resveratrol, and I've shown you different studies. But with sulforaphane, they've done studies with nematodes where they just add a bit of sulforaphane to that feed, the media, and they will have an extended life, significantly extended lifespan with sulforaphane. So sulforaphane, which is found in cruciferous vegetables, so broccoli, cauliflower, Brussels sprouts, etc. Sulforaphane literally has components that goes in, like I talked about the blueberries and things like that, that causes acetylation, so turning certain genes on, involved in detoxification, so phase 2 detoxification, as well as antioxidants such as NQO1, etc. Resveratrol. So there's some, I don't know if anyone knows about the controversy over resveratrol. It is part of our protocol, and most of the studies, well, a bit like the others, yeast models have shown that resveratrol is really, really beneficial for increasing health span and health markers. But again, it's animal models, it's yeast, it's not in humans. There's now some suggestion that perhaps we don't get the same results in humans, but we just don't have a lot of studies in humans. Resveratrol is obviously something that's found in the skins, particularly of, things, grape skins. We think of it with red wine, but also other skins of food. So it can act as an antioxidant. The other one is NAD precursors. So things like NAD, CoQ10, things that are really important for the mitochondria, for energy, vitality. These things decrease as we age. So for example, with CoQ10, by the time we are 65, we on average have half the levels of what we had at 25. So as we age, NAD, CoQ10, glutathione, all these things that help our bodies function really well, help the mitochondria make ATP for all the reactions that need to occur every single day, these decline. So nicotinamide riboside, or NMN, are NAD precursors. They're things that help increase NAD. Ubiquinol is the active form of CoQ10. B vitamins are obviously really important for methylation, so folate and B supporting that methylation pathway. And then omega-3s and vitamin D. These are fairly kind of standard. Most of you are probably giving these to your patients already, but there's actually a lot of research. So some of the more sort of interesting things that we're starting to hear about when we talk about, health span is, NAD, resveratrol, etc. But we probably have the most data around omega-3s and vitamin D. So with our study, as I said, I do have a co-investigator, Paul Taylor. So he's part of the Mind, Body and Brain Performance Institute. And I said to you at the beginning of this talk about resilience. So his actual PhD, so he's ex-military and has been in some very extreme positions, in the things that he's done. So he actually works a lot with resilience, and he's actually doing some research at the moment with the University of Tasmania, which incorporates a lot of what we're doing, but the focus is more around resilience rather than health span. But as I said, they're so tightly connected. So the aim of our study was to determine if a 12-week nutrition, supplement, and lifestyle intervention program can alter DNA methylation and biological age. And DNA methylation is a robust marker of biological age and age-related conditions, as I've shown you. So the hypothesis was that nutrition and lifestyle habits can reduce biological age, measured via DNA methylation, and we've used this true age test. So we have completed the first 3 months, but we've had, of the 60 people that joined, we've actually got 38 that are doing the second 3 months, and Denise has joined us. She's doing her first 3 months while we do our second part. So I'm not going to share the entire protocol with you, but just to give you some indication of the foods, which is obviously really important for us as nutritional medicine practitioners as well. We know food is medicine. So the recommendations are on most days to try to consume at least, or 6 serves of veggies, including 2 serves of cruciferous, 2 serves of greens, and 2 serves of coloured veg. So coloured being purple, orange, yellow, red, etc. It's quite a high protein intake, and this is based on the fact that we are asking them to do some strength training. My system that I have allows people to track food as well. This is not compulsory. They don't have to do that, but we did suggest at the start that people complete a few of these food diaries just so that they can see that they are hitting these targets. A maximum, so people don't have to have this many, but a maximum of 4 serves of whole grains. I think that whole grains are really important for the gut microbiome. And we do suggest, low gluten or gluten-free. When it comes to a serve, half a cup of cooked oats is 1 serve. So if someone's having a big brekkie and they're having a cup of oats, that's your 2 serves. You've really only got 2 other serves if you wanted to have maybe some rice in the evening, things like that. For those, we do have a couple of people that are exercise at a really intense level, the type of people that have been attracted to this study. Some of them, they are, full of muscle and training and in that kind of biohacker world. Some of them may be able to have slightly higher intakes just to make sure they are getting the nutrients they need to recover, replenish glycogen, etc. But for most people, a maximum of 4 serves is enough. Oops. 2 serves of nuts or seeds, 1 or 2 serves of fruit, or if there's 2 serves of fruit, we sort of talk about sort of low sugar fruit, particularly berries. Fermented food, if they don't have histamine sensitivity, and plenty of healthy fats. And it's chopped off the bottom, but we also talk about herbs and spices and using these liberally through cooking. So when it comes to sleep, we recommend 7 to 9 hours most nights. There is some information on sleep hygiene, breathing exercises, meditation, and mindfulness daily, 10 minutes if they can, and aim to move for 30 minutes as many days as possible. So the app, which is actually more designed for the resilience, this is part of Paul's PhD work, has a lot of resources around exercise, mindfulness, and then we adapted this to match my protocol. So I came up with the protocol, and then people can tick off, I've had my supplements, I've done my mindfulness, I've had my serves of veggies, etc. And I don't have it here, but cold showers were also, that was something that Paul really wanted in there. He's a big one on sort of hot and cold therapy. So we did discuss that with everyone as well. So when it comes to supplements, there were 5 products included. So a B complex, I think those of you in Hong Kong can access this B complex. I know you've got it because Denise has it. But for those in the Philippines, if you were interested in supporting methylation or doing something similar, you would be able to get the folate and the B12. So, having those together. But in this case, we've got a B complex with 400 micrograms of methylated folate, 400 micrograms of methyl B12, plus those B cofactors. We have 100 milligrams of transresveratrol, ubiquinol, 100 milligrams twice a day. So that is that CoQ10, an NMN product that has some other ATP support that I'll show you, and a liver support. So I would have loved to have had a lot more supplements in here, but the reality is this is a lot. And for some people, most people are happy to have these supplements, but there are a couple of people, it's a lot to be taking this many tablets a day. So I really wanted to sort of reduce this to what I thought would be the most effective. Obviously, I don't have the results. We've just finished the first 3 months. We're waiting on results. But what I did talk to people about individually was if they needed to have omega-3s. Now, a lot of people were already on omega-3s. That was fine. But I did say to people if they weren't eating at least 2 serves of oily fish per week, and educated them about, having wild and potential, environmental pollutants with seafood and things like that. But yeah, to have an omega-3 if they weren't getting that oily fish. And some of them actually, we've got a couple of people that actually love sardines. We're actually having a very good diet. And we decided that, they're probably getting enough from their food. I think those of you in Asia probably do get a lot more omega-3s than us here in Australia. Some people just don't eat a lot of oily fish. And obviously we want the small ones, not the big ones. Vitamin D, so we did do vitamin D testing, blood testing, and if they were low, adding that in, as well as iron. So we did iron studies as well. So there were some bloods done at the beginning of the study. So this is the particular complex with the NMN. And as you can see, there are also a few antioxidants, as well as some carnitine, a little bit of resveratrol that adds on to the other one, a bit of NAC. Obviously, if you're going to give NAC on its own, you might be thinking more like, a gram or 1,000, milligrams, but some here, alpha lipoic acid and a little bit of CoQ10 as well. So this is something that, when we're thinking about sort of ATP, health span, longevity, this has a lot of the things you need, but obviously it's a relatively low dose. If you're going to have this on your own, you might be sort of having, 2 capsules multiple times a day. In this case, it was 2 capsules twice a day, but if we didn't have all of the other supplements and this was all you were giving to your patient, they might have that a little bit more throughout the day. And then when it comes to the liver support, this is one of my favorite supplements, the liver GI. Actually, not that I feel like I'm totally selling the whole thing now, but I actually have all these supplements on my desk. Not to say to you, oh my God, you should get them. I have them here every day, because if they're not on my desk, I won't have them. So I have been very good and very compliant. I'll be honest, I don't have supplements every single day. When I was recovering from my autoimmune disease, I did. There are certain times if I'm under, higher stress, I might, in winter, I might have some liposomal vitamin C. But, as practitioners, we kind of know what we need when we need them. But I have been diligent with all of these supplements. And I'm also doing the second 3 months. I'm doing this for 6 months to see if I can get even better results. So this is the liver GI, and it has that sort of sulforaphane, which was, something I really wanted, as well as the curcumin. So I didn't really talk about the turmeric or the curcumin when it comes to health span and longevity, but there is quite a bit of research around that as well. So just to give you a quick run through on the study, we had over 300 people respond, and I didn't do any promotions. I just did a podcast, and then I've got a Facebook group and I mentioned it. So that was overwhelming to have that many people want to join, but that shows that there's a big interest in the area. Once people realized, the costs and what was involved, because they needed to pay for the test, all of that, we had about 60 people join up. A few dropped out. Then in March, everyone did their test. This here, where I've put in sort of results complete, these are the first lot of results. So we now actually have all of the results from every single person. I put these slides together a little while ago, last month, but we now have all of those results. And when it comes to the second test, I said here back in August, 30 people had ordered that second test, ready to sort of do it. I think now we have close to 50 people that have done their test, and I've got about 5 people whose test results are back. The issue with the True Age test is it takes about 8 weeks. I don't know how long it took Denise to get, she can tell us at the end how long to get her results, but here in Australia, it gets sent to a distributor. They send it over to the US. It takes us about 8 weeks to get the results. So I've just had my first couple of results come through, and they've actually come through much quicker. We were told at the beginning of the study we would get results in, about 4 to 6 weeks, but we definitely didn't at the beginning. So yeah, I haven't looked at any of those yet, but we do have a Facebook group, and a couple of the people that have seen them, they get uploaded to their portal, have just said that they are, really impressed. And it looks like the first couple we have reverse biological age, but we'll have to wait and see what the results are. And if you are interested, we can come back next year. I don't know if Denise wants to be a case study. I was actually really embarrassed about my results. I thought, I'm the practitioner. I don't want to tell everyone that I've come out older. Like, I actually felt a bit of shame. It was a really strange feeling. I knew I'd be a little bit older. As I said, not making excuses, but I've had Graves' disease. I saw myself age in the mirror. I look at photos before and after, I've seen it. And I was really sick. Without going into my story, if you don't know it, I was, I had to leave work. I was bedridden for a while. I was so skinny. I was just eyes bulging. I became thyrotoxic. It was, I was really, really sick for a long time, and it took me about 18 months to recover. But off medication and didn't go down, that route. I've still got my thyroid, didn't have radioactive iodide and all that stuff. So well now, but obviously that was an impact. And, and many other things. Overtraining in my younger years, working too much, I love my job. I'm so grateful I love what I do, but I'm sure most of you listening, we're very passionate people. We give a lot for our patients, and sometimes we just need to stop and take a moment and think about ourselves. And actually, when I started this study, it was because I genuinely thought at the end of last year, I might have had a flare or got sick again. I was just, a lot was happening last year. And I knew that I was letting go of the self-care and what I need to keep myself well. So sometimes having results like this are the kick in the butt you need. I was thinking we're at the end. Actually, I'll just run over these things quickly. I had some examples to show you the test, but so this is the test, and the aim is if you can be 7 years younger, that's been associated with a 50% reduction in disease rate and age-related conditions. So this intrinsic age at the top, this is like your overall biological age. Think of it like your total body burden, in the fact that this is based on inherited epigenetic patterns from mum and dad, as well as things that happen in your childhood and up until this point in time. So dietary things, exposures, stresses. This is that total body burden, that biological age. The one at the bottom, this is specifically related to immune age. So this is looking at subsets of our immune system. Most people come in about 7 years younger. Our immune cells age at a bit of a slower rate, if you picked up on me saying that earlier. In this case, she's 17 years younger. This is wonderful. But that's more to do with your immune age. And then, the extrinsic age is to do with your immune age. The Dunedin pace is what I mentioned, is really linked with that sort of function. So more to do with health and function, and not necessarily a number. So in this case, this is that same lady. So she has a slower pace of aging. And I will say that even though I came out biologically older, my pace of aging is actually pretty good, is down there. So that shows you that even though I might have experienced certain things in my lifetime that have increased my biological age, when I did this test, 3-4 months ago, probably 4 months ago now, I'm still waiting on my results of the second test. My Dunedin pace was quite good, which shows that right now I'm obviously doing things that are beneficial. We do get an estimate of telomere length, but I will just mention this is not actually measuring the telomeres. This is measuring the methylation pattern on the telomeres, which has been linked to length. So it's like a surrogate marker for telomere length. And then just running through, this was sort of back in August. This could be updated slightly, but just overall, when we look at biological age, that intrinsic age, that total body burden, about half had a lower age and about half were higher, so 28, 27. When it came to the extrinsic age, the immune system, most people were pretty good there. There was a couple of people, and I won't go into it now in the interest of time, but one of these people definitely has an immune issue. And the other, I think, even though he was devastated by it, really, because he thought he was going to come in really like at least 10 years biologically younger, and he didn't. He came out older, and he had the higher extrinsic age, which is a bit rarer. But he's the kind of guy, he's quite extreme. He gets up at like 4 o'clock in the morning and does sprints. Like he's, he showed me his backpack. He's like, I have this backpack full of veggies every day. I juice all these veggies. I do these sprints. I do this. I do that. I mean, there's a point where too much of anything is not good for you. So I'm trying to educate him to pull back. When it comes to pace of aging, about 10 people were kind of equivalent, meaning they're kind of aging at the same rate as their chronological age. So 1 year, every year they get older, they're kind of at a cellular level likely to be getting 1 year older. Most people had a slower pace of aging, so that was nice. And then we had 4. So since I've done this, though, the last couple of results that came in, we had a few delays with a couple of people that had to repeat the test. One person, they got lost in the mail, but we've actually had a couple more with accelerated aging, actually. The telomeres, that sort of breakdown there. And then I took out a few slides in the interest of time, but we do actually get some information linking to epigenetic changes that predict diabetes. So there were some people, and interestingly, most of them have a strong family history. And because I got bloods as well, did have some issues around blood glucose or HbA1c. So if they have a low risk of diabetes, based on the literature, the algorithm, what this is based on, and the science, this is predictive for about 7 years. So assuming they're not changing anything drastically, they're at low risk of diabetes for the next 7 years. We get some information on weight loss. Some of you may have experienced at certain times you cannot lose weight. There are epigenetic components. Methylation patterns can change, and on certain genes that will prevent you, or the body will try to stop you from losing weight to protect you. Yeah, so that's really, I'm loving going through that one. That's really interesting. With smoking, we all had low risk, and alcohol, there were some people that had some methylation changes that are linked with adverse health conditions and alcohol. So in summary, we know that our DNA is important. We have certain genetic variations that influence our health and well-being. But even though we can't change our genes, we can do a lot with our environment to impact that epigenetic, or that epigenome, the epigenetics, and that will impact our phenotype. Our phenotype is what we're expressing, and our phenotype can change. Things like eye color won't change, but if you've got high homocysteine, or you've got type 2 diabetes, or you're overweight, these things can change. You can change, you can get to, and homocysteine is one of the things we measured in our bloods too. But, you can get back to having a, optimal range of homocysteine. You can have optimal, vitamin D levels. You can, at one point you might have a diagnosis of a disease, another point you might be in remission and not have that disease. So phenotype is what you're expressing. So the takeaways for tonight are that age is the #1 predictor of poor health and chronic disorders. But I hope that you can see from this, and you probably already have some fundamental knowledge around this, we can, with the work that we do as functional medicine practitioners, improve health span, improve vitality, and through that too, we are also improving resilience. So epigenetics, specifically DNA methylation, is tightly linked to biological age and cellular health. We know that DNA methylation can be inherited, but it's also influenced and can change with diet, lifestyle, and exposures. We now have a number of epigenetic and aging clocks, and we are in the process of running this pilot study, and I hope to be back next year and share that data with you if you are interested in hearing more. So thank you so much.